Hello friends, good evening. Welcome to SAM Biology class. Friends, today we shall discuss about major histocompatibility complex. Both T and B cells use surface molecules to recognize antigen, but they accomplish this in very different ways. B cell receptors recognize antigen alone, but T cell receptors only recognize pieces of antigens that are positioned on the surface of other cells. These antigen pieces are held within the binding groove of a cell surface protein called as major histocompatibility complex molecule encoded by a cluster of genes collectively termed as major histocompatibility complex locus. The MSC got its name from the fact that the proteins encoded by the genes determine whether a tissue transplanted between two individuals will be accepted or rejected. There are two main classes of MSC molecules, class 1 and class 2. Class 1 and class 2 MSC molecules are membrane-bound glycoproteins showing structural and functional similarity. They serve as highly specialized antigen-presenting molecules with groups that form unusual stable complex with peptide ligands, displaying them on cell surface for recognition by T cells via T cell receptor engagement. In contrast, class 3 MSC molecules are a group of unrelated proteins. They show no structural resemblance or functional similarity with class 1 and class 2 molecules, although many of them do participate in other aspects of the immune response. Structure of class 1 MSC molecules. Class 1 molecules are present on all nucleated cells in the body. They specialize in presenting antigens that originate from the cytosol such as viral proteins. A single class 1 MSC molecule is composed of a 45 kilodalton alpha chain and a 12 kilodalton beta 2 microglobulin molecule. The alpha chain, also known as heavy chain, is organized into three external domains, a transmembrane domain and a cytoplasmic anchor segment. The external domains are alpha 1, alpha 2 and alpha 3. Each external domain is about 90 amino acids long. The transmembrane domain is of about 25 hydrophobic amino acids followed by a short stretch of charged amino acids. And the cytoplasmic anchor segment contains 30 amino acids. The other component of the class 1 molecule is a beta 2 microglobulin, which is similar in size and organization with the alpha 3 domain. This component do not have a transmembrane region and is non-covalently associated with the alpha chain. In a class 1 MSC molecule, the alpha 1 and alpha 2 domains interact to form a platform of 8 antiparallel beta strands spanned by 2 long alpha helical regions. The structure forms a deep groove or cleft with 2 alpha helices as its side and the beta pleated sheet, which is composed of 8 antiparallel beta strands as the bottom. The groove is large enough to bind a peptide antigen of about 8 to 10 amino acids. Then, the beta 2 microglobulin and the alpha 3 domains are organized into 2 beta pleated sheets. Each sheet is composed of antiparallel beta strands. The structure known as immunoglobulin fold, a characteristic of immunoglobulin domain. Because of the similarity, class 1 molecules and beta 2 microglobulin are classified as members of the immunoglobulin superfamily. In this figure, you can see that this is the peptide binding group which is formed due to the interaction between alpha 1 and alpha 2 domains. This binding group is composed of a floor of 8 antiparallel beta strands and 2 antiparallel alpha helices as sites. This group is large enough to bind with a processed peptide antigen of about 8 to 10 amino acids. Structure of class 2 MSC molecule. Class 2 molecules are expressed on almost exclusively a subset of leukocytes called as antigen presenting cells. They specialize in presenting peptide antigens that originate from extracellular space. Those have engulfed by cells such as bacteria. Class 2 MSC molecules are composed of two non-identical glycoprotein chains. A single class 2 molecule is composed of a 33 kilodalton alpha chain and a 28 kilodalton beta chain. The alpha chain is organized into two external domains, alpha 1 and alpha 2, a transmembrane domain and a cytoplasmic anchor segment. Similarly, beta chain is also organized into two external domains, beta 1 and beta 2, a transmembrane domain and a cytoplasmic anchor segment. As in case of class 1 molecules, here also the membrane proximal domains alpha 2 and beta 2 are organized into two antiparallel beta pleated sheets. They form a structure called as immunoglobulin fold, which is characteristic of immunoglobulin domain. Because of the structural similarity, class 2 MSC molecules are classified as the members of immunoglobulin superfamily. The membrane distal domains alpha 1 and beta 1 they interact to form the peptide binding group where a processed peptide antigen of about 13 to 18 amino acids can bind. So this is the peptide binding group which is formed due to the interactions between the alpha 1 domain and beta 1 domain. This binding group is composed of a floor of 8 antiparallel beta strands and 2 antiparallel alpha helices as sites. It is the peptide binding group where the antigen of about 13 to 18 amino acids bind. Now we shall discuss about some of the important functions of major histocompatibility complex. The main role of MSC is to present antigens obtained from different sources to appropriate T cells for recognition in order to destroy the invading pathogens. MSC class 1 molecules are present on all nucleated cells. They specialize in presenting endogenous antigens to CD8 plus T cells, also known as cytotoxic T cells. They play a role in destruction of virus-infected cells as well as tumor cells. Class 2 MSC molecules are present exclusively on a subset of leukocytes called as antigen-presenting cells. They play a significant role in graft versus host response. They present antigens obtained from extracellular sources to CD4 plus T cells, also known as T helper cells. The MSC complex on cell surface is necessary for self-recognition and prevention of immune system targeting its own cells. The MSC molecules play a role in distinguishing own tissue from those present in another individual within the same species. This is known as allorecognition, and it plays a very important role in prevention of successful organ transplantation. Some MSC alleles are associated with increased risk of autoimmune disorders such as Hodgkin's lymphoma and multiple sclerosis. Finally, class 3 MSC molecules are involved in complement activation and in inflammation caused by cytokines and tumor necrosis factors. So friends, this is all about the structure and function of major histocompatibility complex. For more updates, please subscribe my YouTube channel Sam Biology Class. Thanks for watching.